All right, this is the second part to a lecture on virology, really serving as an introduction to this topic worthy of a course unto itself. Here we're going to think about the bacteriophage and plant viruses. The learning goals that I hope that you'll achieve by participating in this lecture are to be able to answer the questions shown here. You should be able to define what a bacteriophage is. You should be able to describe the possible structures of bacteriophage, talk about the two basic patterns that we observe in bacteriophage life cycles, and understand what the common challenges are for bacteriophage and plant viruses with regards to the early stage of viral life cycle, and know the strategies by which these viruses address that challenge. You should also be able to provide at least two examples of very important plant viruses. Bacteriophage are viruses that infect bacterial cells. The typical form that you have probably seen before when learning about bacteriophage in genetics class is the form shown towards the bottom of this figure. And in fact, that is the most common form that bacteriophage achieve. So in that form, we have a nucleocapsid shown here. That means a protein shell enclosing the viral genome. Most bacteriophage have a genome that is double-stranded DNA. However, there are some that contain genomes that are single-stranded DNA or RNA genomes. When found as double-stranded DNA, that DNA is in the typical alpha helical configuration that you are familiar with from cellular organisms. The lower portion of the figure shows bacteriophage that have structures that are known as tail pins or tail fibers. That's what's shown here in purple. And this area would be called a sheath. This figure depicts specifically the bacteriophage that are known to infect the bacterium Escherichia coli or E. coli. Because bacteriophage were discovered relatively early in the history of microbiology, specifically in 1915 by Frederick W. Twort of Great Britain, and then 2 years later in 1917 by Frederick Derel of France, we've known about them for a very long time, and we've been studying them very carefully for a very long time. They're relatively easy to grow because you only need to be able to culture bacteria as the host cell. And this is often, at least in the case of E. coli, easier to do than culturing plant or animal cells. This makes bacteriophage really useful models for studying viruses in general. A lot of our knowledge of virology is based on studies of the bacteriophage, especially bacteriophage that infect really well-studied bacteria like E. coli. So let's think a little bit about the life cycles or the replication patterns that we see in the bacteriophage world. And you should have seen a diagram similar to this in your genetics class previously. So when we think about bacteriophage life cycles, there are a few terms that we need to learn. And let's start with the terms lysis and the terms lysogeny. Okay? And let's remember that there are different definitions or descriptions for bacteriophage. One of the key ways that we distinguish one type of bacteriophage from another is to say that a bacteriophage is either virulent or temperate. You need to remember that virulent bacteriophage always use the lytic pathway, which is shown over here on the left in this figure. That's all they can do, all right? In contrast, temperate bacteriophage are bacteriophage that can switch between the lytic and the lysogenic pathway. Now let's consider these two pathways. Let's start at the top and let's just look. Here is the host cell, right? So this is some bacterial cell. Bacteria are the host cell. Here's a virus. This is a bacteriophage. In this cartoon, because they're showing us both pathways, they're showing us a temperate bacteriophage. So the virus attaches to the host cell, and that's a very specific attachment as I discussed previously. The genome of the virus enters the host cell, and here it's shown in darker green. And then let's assume that we're proceeding down the lytic pathway, which is all a virulent bacteriophage could do. Once the genome of the bacteriophage enters the host cell, then what will happen are the phage or viral components will be synthesized. So in order to do that, you're going to have to make more viral DNA, and you're going to have to make more viral protein. So once you make more viral DNA and more viral protein, which is happening in these steps here, then you have enough new viral or phage components to assemble new viral particles, also known as virions. So these are multiple new viruses, okay? And they're increased in number relative to the original infecting viral particle or virion. Once you have a lot of those assembled, you actually build up so much pressure inside of the host cell that it will lyse or burst. And that's what's happening down here. And that will release all of these many new phage virions, and they will go off and they will find new host cells and they will attach and they will propagate or continue this pattern or this life cycle. Now remember, if you are a virulent bacteriophage, that's all you can do. So every infection event results in death of the host cell. Okay, those are the kinds of bacteriophage that we can track or count using the plaque forming assay that I described in the previous lecture. Now that is not the only possibility in the world of bacteriophage. So in the bacteriophage world, there are many different viruses, many different bacteriophages that are actually temperate. A temperate bacteriophage can use the lytic pathway, and that's how they get from host cell to host cell. But they can also use what's called the lysogenic pathway. They do what is called lysogeny. Okay, that would be the noun. So the lysogenic pathway is shown over here on the right-hand side. So here we have, back up at the top, we have the virus attaching to the host cell, just like before. We have the viral genome entering the host cell, but then something quite different happens. Here's the viral genome, okay, and here is the host genome. And what's happening is integration, all right? The viral DNA is actually integrating into the host cell. When that happens, the term for the virus is called prophage. So at this point, instead of saying virus or virion, we say prophage, okay? And prophage means that the virus exists only as a piece of DNA integrated within the host cell genome. So if you were to measure the size of the host cell genome at this stage, when it has been lysogenized, it is now larger and it now carries viral DNA. And that means that every time the host cell replicates in kind of the normal process of DNA replication, 2 new chromosomes followed by binary fission, and then complete cell division, whenever that happens, the viral DNA is replicated. So the viral DNA is being propagated or increased in amount at the same time that all the rest of the host DNA is being increased. Now this can have some pretty profound implications, okay? Because that viral DNA shown in here, right, it could be acting as a potential mutagen. It could be conferring new genetic properties, and I gave you an example of that in a previous lecture. I explained to you how the cholera toxin protein is encoded by a gene that is actually viral in nature. And so in Vibrio cholerae, it's pretty much only a pathogen when it is a lysogenized cell. That's actually part now of the biology of Vibrio cholerae. So the genetic and the physiological impacts of lysogeny are quite widespread. And over time, it seems like the lysogenic pathway that can be undergone by many temperate bacteriophage, or by all temperate bacteriophage, is a really important aspect in creating more genetic diversity within host cells. So it's a favorable situation overall, and that probably explains why temperate life cycles are so very, very common. Now, of course, at some point in time, as I said before, temperate bacteriophage can switch back to the lytic pathway. What triggers that depends on the virus and the host cell that you're studying. It might be triggered by differences in culture conditions or other environmental signals, but they can go back to the lytic pathway. That's called induction when it happens, and it's shown here in this particular figure. We will talk a little bit more about the implications of bacteriophage life cycle on the process of horizontal gene transfer in future lectures. In an earlier lecture, I already discussed how bacteriophage will very specifically adsorb to their specific host cell. So I won't repeat that here, but I will point out that this is bacteriophage. This image is based on bacteriophage T4, and it is infecting its chosen host cell, which is Escherichia coli or E. coli, a gram-negative bacterium. You're looking at that gram-negative bacterial cell wall here. So the T4 bacteriophage is adsorbing or binding to the outer membrane and associated proteins in E. coli. That's what's happening in A. The tail fibers are attaching firmly. They're the purple structures shown there. And then what's happening is the sheath, which is this part here, okay, is contracting. And when it contracts, what happens is there's an injection of an enzyme called lysozyme. You've heard of that enzyme before. It's an enzyme that we also produce, although we make a slightly different form of it, and we put it in our tears and our saliva. It's an antimicrobial enzyme because it allows for the degradation or breakdown of glycosidic linkages found in peptidoglycan. So this is a viral enzyme. It's carried within the viral nucleocapsid. Actually, more specifically, it's in the sheath, and it is being injected, and it is degrading the peptidoglycan. And that creates a hole, which is shown here. And once that hole has formed, then you have penetration of the cell wall, and you can have the injection of the viral genome, which is shown in green here. So this is the particular way that T4 bacteriophage deals with breaking through the cell wall. Use of enzymes is a very common pattern in bacteriophage as a means of degrading the cell wall sufficiently to allow injection of the host cell genome within the host, or into the host. So we can't really talk about bacteriophage without talking about restriction enzymes. As we talk about them and as you consider them and you read about them in your textbook, I don't want you to think that the bacteria themselves are completely defenseless against this constant viral assault. Over time, all host cells have evolved or co-evolved with the viruses that infect them, and that's true no matter what life forms you're discussing. In the case of the bacteria and the archaea, they have evolved to produce enzymes that are called restriction endonucleases. Okay, the term endonuclease means an enzyme that will attack a nucleic acid from within. Okay, so not from the ends, but from within. Shown in this figure is EcoR1. It's made by E. coli, and it's the R1 restriction endonuclease. It binds very specifically to sequences on double-stranded DNA that look exactly like that, and then it cleaves that double-stranded DNA, so it will break that. And you might be wondering now, well, why would the bacteria make an enzyme that can degrade DNA? Isn't that a problem for the bacterium itself? And the answer to that is that the methylation patterns differ in viral DNA than in bacterial DNA. And so the EcoR1 restriction endonuclease and other endonuclease enzymes are able to identify these sites and only in viral DNA. And when they identify those sites, they will bind to them and they will cleave it, and that will destroy the viral genome, which will protect the host cell against viral attack. We take advantage of restriction endonucleases in the lab. We can purchase them commercially, we can make them and sell them commercially, and we can use them to cut up DNA for laboratory purposes, so they're kind of part of the overall biotech industry at this point in time. But they are a product of nature. There's something that bacterial and archaeal and organisms make to protect themselves against viral attack. So enough about bacteriophage. Let's consider for a moment now plant viruses. And I'm not going to say a lot about plant viruses, even though they're really incredibly important for a lot of different reasons. And one of the reasons I won't say a lot about plant viruses is that we actually know less about plant viruses than we do about animal viruses or bacteriophage. I would say this is an area of growing importance, and that there are going to be lots of opportunity for careers in that particular area. And one of the challenges that plant viruses have, that is the same challenge experienced by bacteriophage, is that they have to deal with a host cell wall. Plants, like bacteria, have cell walls. And so plant viruses have to attach to their host cell, then they have to get across the cell wall in order to get inside to replicate. Shown in this particular image is just a schematic showing plant cells. Plants are multicellular organisms, so they have cell-cell attachment points that are very, very important and specific. And between cells, they have very specific channels called plasmodesmata, and they allow only certain substances to go back and forth. One of the strategies that some plant viruses have dealt with is to take advantage of the plasmodesmata by using them as entry points to get into the host cell. So there are movement-associated viral proteins that some plant viruses have that essentially force the plasmodesmata to open, allowing viral passage. That's one of the strategies that some plant viruses have evolved as a way of getting access to the host cell cytoplasm. There are many other ways that plant viruses can penetrate host cells, however. For example, any sort of mechanical injury that breaches the cell wall and therefore transiently breaches the plasma membrane of underlying cells will allow a virus to enter. This could be injury due to arthropods, insects, or nematode activity. So a lot of plant viruses are carried by insects or carried by arthropods or nematodes. And when those animal creatures damage the plant cell wall, then viruses can enter the plant cell. Viruses can also follow in the wake of fungal parasites, which might burrow into the plant cell wall, leaving an opening for viruses to then come through after. And another interesting thing that has been observed is that plant viruses can be vertically transmitted through infected seeds or vegetative propagation. So during agriculture, if we graft infected tissue onto healthy tissue, we, as those plant tissues grow, we see vertical propagation of some plant viruses. These are just a few of the many strategies that plant viruses use to help them penetrate host cells. I'm going to leave you with a little bit of homework. I'd like you to come up with a list of important plant viruses, and I prefer that you came up with a list of economically important plant viruses, but you could come up with another list as long as you have a reason why. An example of an economically important plant virus to get you started is the maize streak virus. This is a plant virus that is endemic in sub-Saharan Africa. It's transmitted by insects and infects corn. Corn is the major crop in that region. And it causes plant stunting and crop loss. So it's accountable for at least, in some years, 10% loss of the overall corn crop, which people are relying on to eat. And so it's the propagation of that particular plant virus is directly linked to human hunger and in some cases starvation. I'd like you to read the blog posting at the site indicated here and create a list for homework of 5 important plant viruses, and we'll talk about that in class. I'll also be checking your notes to see that you have that list. And that concludes this lecture.